Okay. Now, let us briefly take a look at the autonomous nervous system. I will give you some thumb rules, okay, which will hopefully make your life simple. One thumb rule is that parasympathetic nervous system, which generally post ganglion fiber, post ganglionic fiber releases acetylcholine. Acetylcholine is generally acetylcholine is there post ganglionic, of course, in parasympathetic. Several of the neurons of the myentric plexus also the neurotransmitters are same. There is acetylcholine, there is epinephrine, there is norepinephrine, there is serotonin, there is bradykinin, there is dozens of them, and then there are peptides about which I am not talking. But my point at the moment is, I am here to give you some thumb rules. One thumb rule is that acetylcholine generally stimulates the smooth muscles of the alimentary canal, exactly opposite to the cardiac muscle, and it works at extremely low concentrations, 10 raised to the power of -5 m. Are you with me? If you prepare acetylcholine solution, okay, and then if you drop just a drop on the smooth muscle of the alimentary canal, it contracts. I am saying this because I have done it. Okay. Okay. And you know something, and then what experiment we would do is make a solution 10 raised to -5 and then see how much is the contraction, then 10 raised to -4, which is 10 times concentrated, and then see whether you get a, and then 10 raised to -3, and then draw a dose response curve. Okay. Standard M-Pharm practical. Okay, when you are a student in pharmacology, this is standard practical. Then what you are really doing is you are assaying. It is a bioassay, get the language, it is the bioassay of acetylcholine in your given unknown solution. That is your problem, okay? And you can find out the concentration using a biological method, okay? Because a chemical method may not be as sensitive, and a biological method is because that acetylcholine is really working, okay? Good, good, okay. So, here we are looking at the spinal cord, central nervous system, here is the medulla oblongata, and from medulla oblongata you are getting a parasympathetic nerve, and I do not have to tell you that we are talking about vagus. What are we talking about? Vagus, okay. So, this vagus author has not shown, it will be preganglionic, postganglionic, and it acts on here, he just see where it acts, all the way, vagus, branches of vagus. Done so far? Okay. And then from the, it's a, what is the other name of parasympathetic nervous system? Craniosacral, you are forgetting. What do you call it as? Craniosacral. What do you call it as? Do not forget, craniosacral. Sacral. So, from the vertebral column. What did I say? Say that again. Vertebral column. Say that again. From the vertebral column at the level of sacral 2 vertebra, at the level of 3 vertebra, at the level of sacral 4 vertebra, the nerves will come out. And those nerves will go where? Those nerves will go to the horizontal colon, to the descending colon, and to the rectum. Okay. So, this is the parasympathetic nerve supply to the alimentary canal, and this will and if this system is if that system is activated, if this system is activated, it will promote peristalsis. Okay. It is excitatory, it is generally excitatory, and not only it excites the smooth muscles, it also excites the various glands, because you will need to secrete juices, containing enzymes. Okay. So, it is not only the smooth muscles, but also the gland. Okay. Now, I am going to ask you a question, okay, and let me see how smart you are. Before operating on a patient, okay, a doctor or the anesthetist may operate, okay, an agent which will block the action of acetylcholine, or a substance that will block the parasympathetic nervous system for a few hours. Why? to prevent the secretions, whether they are in the air passage or in the secretion, you want to prevent the secretion, because when you are awake, you see secretions we do not realize, but we care of we make sure that our passages are clean and open. The patient in general anesthesia may not be able to do that. Are you with me? Yes or no? You see, we do not realize if something I will swallow, okay, I'll clear my throat. The patient in anesthesia may not be able to do that. So, the one way is to overall reduce the secretion. How do you reduce the secretion? Block the parasympathetic nervous system temporarily for, okay. So, I will take you now to the next slide, and here we talk about the sympathetic nervous system. So, where are we? We are looking at the vertebral column from what level to what level, please? Say that again. T5 to L1. These are the preganglionic fibers. These fibers which go to the alimentary canal, they generally do not synapse in the ganglionic chain. The fibers just pass through and they enter into a ganglia, which we call as what? They are all sympathetic ganglia. These are all sympathetic ganglia. Are they part of the chain? Are they part of the chain? No, they are not a part of the chain. They are not a part of the chain. Okay, so every sympathetic preganglionic fiber does not enter. It does not end into the chain. It can just pass through and it enters into these three ganglia: celiac, superior mesenteric and inferior mesenteric ganglia. And here you have the preganglionic fiber and the postganglionic fiber which supply. And again, what is the neurotransmitter being used here? Acetylcholine. Excellent, excellent. Everybody should know. What is it? Preganglionic is what? Postganglionic is what? Norepinephrine. What is it? Norepinephrine. So, norepinephrine is being released at its target. Now, where is the target is an interesting question. It is going to the myentric and it is going to the myentric and submucosal plexus. Let me talk about this diagram. This is the epithelial cells of the alimentary canal. Okay, so far. Here you have the lumen. Here the food will pass this way. Okay, so far. And the author shows one network of neurons, and I will call this network of neurons as what? You already know the name. Say that again. Where is this located? Where is this located? Just outside there is a layer of longitudinal muscles, and just inside there is a layer of circular muscles. Okay. And these muscles, by the way, we saw them, right? Yes or no? Yes. How dare you say no, or how dare you doubt I have shown it to you. Okay. We have seen alimentary canal. Okay. There we did not see the plexuses because you need to use a specific stain. Okay. They are there, we do not see them because they are not, we did not use the proper neuronal stains, but they are there. Okay. So, there is a myentric plexus here and there is a submucosal plexus here, and can you see here author has a very interesting way. Just see sympathetic there, mainly postganglionic. Okay. Mainly postganglionic means, if I take you to the previous slide, if I take you to this slide, see these are the preganglionic and these are the postganglionic. So, can you see these red fibers here? Now, keep this in your mind, keep it active, and these are the ones which are terminating on what? On the neurons of the submucosal plexus. Are you with me? Yes or no? It is ending there. Okay. So, this is how the sympathetic nervous system is innervating the neurons of the enteric nervous system. Which component of the enteric nervous system? Submucosal plexus. Done. At the same time, there is also input from the parasympathetic nervous system. They are preganglionic. The fibers are what? So, those fibers which are coming right over the okay. Those fibers, these fibers, they are preganglionic. Okay. They are coming via vagus. The cell bodies of these neurons is located in the medulla oblongata, maybe in the vagal nucleus. Those neurons sitting in the medulla oblongata, they give rise to axons which come out of the brain by way of the vagus, cranial nerve, travel all the way to the alimentary canal. Which part of the alimentary canal that we have seen in one of the previous slides? And then they come all the way and they innervate what? Neurons of the myentric plexus. Okay. So, this is how sympathetic and parasympathetic nervous system is, and then eventually it is these neurons which will by way of now, whereas we know that this guy is going to release acetylcholine, and this is postganglionic is going to release norepinephrine. These neurons myentric and this, I told you once, I will tell you again, dozens of different neuropeptides, neurotransmitters. So, they can have glutamate, they can have GABA, they can have acetylcholine, they can n number of. So, the control of smooth muscles of the elementary canal is extremely complicated. Extremely complicated. It is very delicate control, okay? And it has to continuously get the signal. See, you see, it has to perform a mechanical function of pushing the food. Okay, so it has to know whether the bolus of the food is there, or it yet to arrive, or it has to be pushed how quickly, how far, and then as it is being pushed, has it been properly digested? Has it been depending on the content of the bolus of food, depending on the type of the food that you have taken, okay, more in protein, more in carbohydrate, you need to give the enzyme, you need to give the right enzyme, and most important, how it does I still do not know, but you have to give the right enzyme in right amount. Hello, how do you decide the amount? Okay, I mean, you have taken 25 grams of a particular type of protein today, and 25 grams of another type of protein day after tomorrow. Are you with me? So, the stomach has to decide as to which enzyme I am going to release, in what proportion, and day after tomorrow if you take 50 grams of, then I have to release more enzyme. Okay. So, for n number of computations which the system has to do, okay, therefore, the gut has been provided with a independent nervous system with extremely complicated neurons which I do not understand, okay, so that the nervous system can deal with these extremely fine computations with reference to how to push the food, when to push the food, what enzyme, how much of enzyme, and at the same time sensing the environment. Hello. Sensing the environment means what? After all, how will the system has to know whether the pH is okay or not? Hello. And the stomach has so much pH, okay, and then the chyme which is there at a certain pH, which may be too strongly acidic, into the stomach. Then as it goes into the duodenum, you need to neutralize it, and make sure that you bring the pH to 7, okay, of the food that is no. When for doing that, how much of alkali you have to release bicarbonate ions? How much of bicarbonate ion are you going to release? I mean, it is very easy for me, but how much? Okay, and you have to just release enough bicarbonate so that the pH goes to 7, not that it goes to 7.2 or 7.4. So, you have to release bicarbonate so much. So, which means we have to have a system by which you keep on carefully monitoring the pH of the stomach, so that you can titrate it to the right level. Are you with me? What I am doing is I am not giving you any clear concepts, because my concepts are also not clear. What I am trying to tell you is to give you an essence of the complexity of the situation, okay, so that you will be able to broadly appreciate, accurately it is very difficult, broadly appreciate the importance of the enteric nervous system, which many people also like to call as gut brain. What do you call it as? Gut brain. What do you call it as? Gut brain, because it is very large, and it is roughly estimated that the number of neurons that you would encounter in the gut brain is almost as much as the neurons in the spinal cord. So, your spinal cord has a large number of neurons, and almost an equivalent number of neurons you will find in the gut brain. Okay.
How my elementary canal is able to identify what kind of food I am taking? What type of?
Okay, we are looking at the same thing. Okay, I put this slide because it tells us more about the sensation. Okay, so there are. So let us see. This is a secretory cell. What secretory cell? It might be secreting acid in the stomach. It might be secreting bicarbonate in the duodenum, or it may be secreting a particular enzyme. Okay, so far, and it is being controlled by what? It is being controlled by this neuron. Okay, this neuron is it is being controlled by this sympathetic neuron. Okay. Then it is also being controlled by this neuron. Where is this neuron? This neuron is of the submucosal plexus. Okay. And then the author tells us there are sensory terminals. Okay. These sensory terminals, they will pick up the information about what enzyme, what product of enzyme. Okay, all that information it takes to this neuron. From this neuron it goes to this neuron. From this neuron it goes, the information goes over the vagus. Okay, and the information over the vagus will go to the medulla. I am sorry, to the medulla. Now medulla is processing. Now medulla knows, okay, as to what kind of food is where and what is the pH, and then it will again give back information over the motor fibers over the vagus, which will bring about the motor. No, I am sorry, there is the where are this is their mechanoreceptor endocrine cells. The information will go to the smooth muscles or to the secretory muscles, and the system will respond accordingly. Can I go ahead now? I need to.
So the parasympathetic innervation, they never form a ganglion outside the central nervous system. They directly come from the vagus.
Oh yeah, it does.
They don't form a ganglion.
No, they don't form. The answer to your question is here. Okay, this is a preganglionic. This fiber is preganglionic. Preganglionic means what? Its cell body is in the medulla oblongata, and it is terminating on these neurons. So this is something like a postganglionic fiber. We don't call it a postganglionic fiber. We call it a neuron of the enteric nervous system. But this is so this particular neuron will have receptors for acetylcholine. Okay, and what neurotransmitter this particular neuron contains, I don't know, but author gives us a rough idea of. Okay. Yeah.
Is there any significance of it being preganglionic or postganglionic? Because whatever anyway, only acetylcholine is going to get released.
Acetylcholine is going to get released.
So
Okay.
it's an anatomical feature.
No. Let's not oversimplify. Ask your question again. I don't know. I can try. Yeah.
Is there any significance in it being a preganglionic fiber as opposed to postganglionic? Because either way, acetylcholine is what is going to get released.
Okay. Oh, no. It is not a question of only the neurotransmitter. It is a major question of processing. A lot of processing is happening in the ganglion, okay, which is necessary for autonomic regulation. It is the processing. Is the word processing clear to all of you? Okay, and the information goes to neurons, and then the neurons process and tells, okay, I am going to control that particular muscle, I am going to control that particular secretory gland, and that one, and not that, and all that processing happens in the ganglia, okay, which does not happen in the case of our somatic system. Here it does not happen, but in the autonomous nervous system you need a lot of, means as if in the autonomous nervous system, this is my language, that the nervous system has said, okay, I am not going to take the responsibility of controlling everything at the level of the center. Okay, I will delegate that responsibility to the ganglia. Are you with me? So the ganglia do, and then of course, and then they use the neurotransmitter. So neurotransmitter there is a particular is only a tool. Okay, but the importance of the ganglia is a huge amount of processing. To tell, okay, it is to tell as to ultimately at the end organ how much of neurotransmitter is being released and where it will be released, and all that processing happens in the autonomic ganglia. Let us see, here is his catalog, the catalog of, let us see, what is it? Number 1 acetylcholine, number 2 what? Norepinephrine, number 3 what? We have done this, adrenoceptor, they are very familiar. What is 4th one? 5th one? 6th one, cholecystokinin, 7th one substance P, what is 8th? 9 somatostatin, leuenkephalin, metenkephalin, bombicin, and the list is incomplete, list goes on and on and on. Okay, and then there are and look at the last sentence. The author raises his hands. What is the last sentence? The specific functions of many of these are not known. This is just a different preparation of the smooth muscles, where the people have used different antibodies against different these neurotransmitters, and you can see the beauty of at least they are showing, the author is showing 3 orphanin, enkephalin and calretenin. These are some of the signaling molecules which I just put. Okay. Okay. This is peristalsis. Okay. So obviously, this is a bolus. The bolus, the immediately behind that. Now, please remember. See, whenever we have to look at these images, and we look at the motor component, and we understand immediately, and we feel happy about it. Okay. Okay. But that is only 20% of the truth. 80% of the truth is you should ask yourself, how does the system know? And for that, then you must ask, where is the sensory component? And then how does the sensory component know? And then you will know how hard it is to figure out everything. Are you with me? Means what? It is when the food is here, whereas these muscle cells in front of the food is going this way. These muscles, they need to relax, particularly the circular muscles need to relax, whereas these guys need to contract. Okay, and it's amazing. How do you know with how much force to contract? Are you with me? You see, depends. Whenever we push a thing, we push or lift anything, we immediately get an approximate judgment as to how much strength am I going to need, and exactly equal as necessary. I just apply the necessary amount of force. Okay, so obviously those muscles also need to. Okay, you can't overdo it, you can't underdo it. You just have to do it, and also do it at the right pace. Okay, and for instructing those smooth muscles, you have to have a sensory input. Where does the sensory input come from? Okay, I am not giving you the answer. I don't know the answer. Okay, I am just sensitizing you to this component of the physiology. Okay, so very interesting slide. Look at this. Very interesting slide. I will tell you why. Here, author gives his imagination to tell us as to how actually peristalsis may happen. Okay, so far, for our convenience, author has removed everything, only kept the longitudinal muscle layer, the circular muscle layer, and the myentric plexus. Everything else is removed for our understanding. Okay, so far, we are good with the story so far. Okay. Now, the next thing author does is, author has drawn different neurons and given them different colors, and here is the legend. Okay, and the green one is the sensory neuron. So, what do we have? Author is all his imagination. There is a neuron. Where is it? It is there in the, and the neuron is really there. Okay, neuron is really there. We are imagining its function. This particular neuron here, okay, once as long as the food was here, okay, the food was here, the food is being pushed here, it will go here. Look at the big arrow. We will call this as the bolus of food. As it has gone, so as long as the bolus was here, okay, this neuron was under stretch. Are you with me? Okay. Now, as the food moved forward, the stretch has gone. As a result of that, this now neuron is taking a signal along with the arrow. This neuron excites this blue neuron, which is an interneuron. This blue neuron talks to this brown neuron. Okay, brown is all my artificial colors. Okay, that brown neuron has one branch which goes and stimulates the longitudinal muscle, and it has another branch that goes and stimulates the circular muscles, which are immediately behind the bolus. And when they are excited, look at these arrows, they will press and they will push the bolus in one direction. Are you with me? So this is how perhaps the signaling is happening, so that I want you to spend more time and try to understand what are the different functions that might be undertaken by this slide. I'll stop now, and we'll see about this in the next class. Okay.